All right, good morning, everyone. Welcome to ASRM 2024. My name is Nick Campion, and I'm Global Product Manager with Cooper Surgical. And on behalf of Cooper, welcome to my talk about the ZyMōt multi-sperm separation device. Now, I'm sure many of you are familiar with it. Many of you may use the device. I'm sure some of you still are curious as to what it's capable of and what it can mean for your lab. So that's what I intend to talk about today. I'll begin with a set of slides that will kind of cover the comprehensive nature of the ZyMōt device. Some things are not quite as obvious as others until people use them repeatedly and really see all of the multiple values that ZyMōt can bring to any organization. Some people may hear about us from the low DNA fragmentation that with the sperm that result from ZyMōt. Some people like the fast, easy nature of it. We're going to talk about all this in a comprehensive way. An overview of all things that converge when using the ZyMōt multi-device. So, as we can see, ZyMōt Sperm Separation Device is surrounded by some very comprehensive value points. We'll start at the left. We have fewer steps involving ZyMōt. You'll see this. It takes 3 active steps followed by 1 inactive incubation interval. So total hands-on time is very, very minimal, a few minutes. We also because we have fewer steps, we are reducing potential for error. We're lowering risk. There's a shortened chain of custody that really, those two things work together. So there's more confidence in the lab and less opportunity for potential mistakes. We're innovative because we were the first sperm separation device around. And like I said in the beginning, the questions were always, what is this thing? What does it do? Does it work? And now, after many, many years of labs adopting our device, people come up and say, we love it, or at least the questions are more critical thinking type. And that shows me that people are engaging with the device and that the device is itself doing great things for labs. It's simple to use, which is kind of another way of, you'll be able to see all this in just a moment when I do a demonstration. But also it's highly scalable. So the more people use ZyMōt, the more we see time savings add up, and the more the workflow overall improves. I mentioned low DNA fragmentation earlier. This is the principal clinical benefit of our device if we want to look at it that way. So ZyMōt, based on how it works, which we'll go through, provides us with a very clean sample, highly motile, often in the upper 90 percentile for motility, but that also being combined with incredibly low DNA fragmentation associated with any of all these sperm on average, which is something, of course, we cannot see with our human eye. And of course, we have ways of quantifying that often take time and money for patients, only to be left with confirmation of a problem that we need to find a solution for. So the lower DNA fragmentation associated with ZyMōt sperm is the first thing everyone always validates in any studies, and I'm proud to say that across the board, it's incredibly impressive. So again, simplifying, standardizing, these are keywords. This allows for potentially more people in the lab with potentially less experience to use ZyMōt in an equivalent way to someone with much more experience. So there's flexibility that is somewhat inherent with the device. Again, this is going to go really fast as far as the actual hands-on time, but that is always welcome in the lab. People will have this incubation interval of time to go about their business and do other things if they need to do, or perhaps process another semen sample. So this slide is just it's more of an overview of what I'm going to show you eventually. But as far as the construction of the device, it's a 2-chamber device. We have the lower chamber of the device that corresponds to the inlet ports where we will be loading raw semen into. Or if we have insufficient volume, semen plus media. But it's nice because we, using the raw sample saves time, of course, but also, there's no added ROS generation or any exacerbation of DNA fragmentation inherent in the sample to begin with. Once we have the device loaded, we overlay media. We overlay sperm wash in most cases. Origio sperm wash works great with this, and that upper portion has the media overlaid, and separating the upper and lower chambers of the device is a microporous membrane. Now this membrane has very, very tiny micropores etched into it, and via a particle accelerator, and they are 8 micrometers in diameter, which turned out to be, when the device was developed, just the right kind of Goldilocks size to allow sperm an opportunity to find their way through if they're incredibly progressive in their motility, but also it presents somewhat of an obstruction or a challenge for those sperm that do not display those same qualities. And once we have the device loaded, we simply move on to incubating it in a petri dish and go about your business. Like I said, take a break, or maybe see a patient, or tend to something else. But we know 30 minutes is this convergent point where we can expect the highest yield of sperm resulting from ZyMōt, but also retaining that very low level of DNA fragmentation. And then once that 30-minute interval elapses, we simply remove the device from the incubator and aspirate our final separated sperm sample. All that we need in order to operate ZyMōt is, of course, the device itself. We will also need a semen sample, and depending on our incubating conditions, sperm wash in most cases, an incubator, which we don't have in the picture here. We need 3 1 milliliter syringes, lure slit tip syringes. They work the best. A petri dish and some sort of cat tube if we need to save the sample for later in the day, whether it's ICSI or really any moment like that. So this is an illustration just kind of summarizing what I basically just said. As far as loading the device, takes a few minutes or maybe even less. We incubate, we come back, and we have a hypermodal sperm sample in the majority of cases. And again, DNA fragmentation being the one thing we can't see. And all of this works together to compound in so many positive ways within the lab. Again, shortening chain of custody, lowering the potential for risks that could possibly be made. So a little more on DNA fragmentation. This study was done actually many years ago at this point, and I saw the author walking around earlier. I hope I could say hi to her at some point. But she was curious as to, comparing ZyMōt to other more conventional sperm separation methods. And in this case, she had 30 patients of all different walks of life, of all different ages, of all different starting parameters, from motility to concentration, all of these things. And we split the samples. There's enough for each patient to split between these multiple methods. So we had traditional density gradient of one brand, followed by a swim up of the same brand, and another brand of gradient followed by swim up, and then finally ZyMōt. And the actual graph the data table is really, really impressive, but we wanted to make it a little easier for it to see here. So as you can see, or hopefully you can see, I'm going to move over here for 1 second. This little black bar represents the resulting final DNA fragmentation of the patients in this study. And it was all over the place to begin with, which is another good point, because again, in order to confirm that someone has elevated DNA fragmentation or any type of general male infertility, oftentimes we've already gone through a cycle and had a failure, or we just don't know what we're getting out on the other end. So at least we, with this device, you have that added assurance that it's really going to be the highest quality sample that we can achieve in an easy way such as this. Now, here's this will also look familiar, but what I really love about it is this was unsolicited feedback on one of the international IVF, the I3 as they call it, the International IVF Initiative. And ZyMōt tend to come up with a lot of andrology-based topics. And in this case, Dr. Caroline McCaffrey from NYU wanted to just talk about, at a brief point in her presentation, her experience in using ZyMōt in her lab, which, as we know, is a very well-respected, high-volume lab. And these are the points that she highlighted, to my delight at least. Again, unsolicited. We had no relationship with her with this other than that she used our devices. So improvements in workflow and productivity, hands-on time savings, consistency regardless of skill level or experience, highly scalable, clean samples, and lastly, low DNA fragmentation, which lines up exactly with the value points that I have been talking about, and I'm sure many of you will realize when using ZyMōt in your lab. So for now, I'd like to go through the actual hands-on process of using the ZyMōt Multi 850 microliter device. Again, I have my semen sample. It's water today just to keep things hygienic over here. Gloves are just for show until my big fingers don't distract anyone too much. We have our Origio sperm wash. Again, it works great with this. You may notice also I have our eyewitness tags on the sample, on the device, on our eventual tube. So those are very easily integrated with ZyMōt as well. The Cooper lineup, the andrology lineup in general, ZyMōt is the newcomer, but all of these products work in tandem and are a very robust set of options for any lab. So to begin with, we just need to confirm that our sample has come to liquefaction as best as it can, and that we have at least 850 microliters. So another way of saying that is 0.85 milliliters, which in most cases we probably will have. So we reach for the first of our 3 syringes for this first step. Now again, these are nothing fancy. They're just simple 1 milliliter tuberculin syringes with a lure slit tip, and our device was designed to accept these syringes so that we achieve a very snug fit and a tight seal. So this works to our benefit for a lot of other reasons as well. So we'll begin with our semen sample, just giving it a brief stir, and we will pull up the 850 microliters of the sample that we need. And I'll set this out of the way for now. Now for audio-visual reasons, I have the device facing this way, and I'm going to try to do this in reverse, or kind of upside down in my case. If anything, you'll just realize how much easier this can be to use. So we have I'll tilt it for now. We have our inlet port in the center of the device here. We have an outlet port in the upper left corner of the device, and again, this polycarbonate membrane, microporous membrane separating the upper and lower chambers of the device. So we begin by placing the syringe into the outlet port. I don't recommend doing this in the lab, but just for demonstration's sake, you can see we have a nice snug fit. This really could be an advantage for viscous samples that might be a little difficult to manipulate. You will not experience any upwelling or backflow of those types of samples with ZyMōt. So from here, we just simply begin by plunging down in a slow and steady fashion. And we have some visual feedback as underneath the membrane itself, you can see the sample loading. We do want to make sure that we successfully deliver all 850 microliters of our semen sample into the inlet port of the device. So that is step 1. really only a few seconds. Step 2 involves the upper chamber of the device. It also involves our sperm wash media. So again, we have our original media here, and we will reach for our second of our 3 syringes for this second step. And this second step involves 750 microliters of our media. That's another way of putting that, 0.75 milliliters. Of course, our instructions are very explicit, and there's no need to try to log these volumes away in your head right now. But we will take our second syringe, and eventually we're going to just overlay this media on top of the membrane, avoiding touching it, of course. We have a microenvironment here with very small cellular forms, so we don't want to be stirring up things or agitating the situation. So we begin in the outlet port simply to prime it so that we don't experience any dry spots or surface tension later on when we are going to finally aspirate our separated sperm sample. So once we have primed that port, we just continue around in a circular fashion, droplet by droplet, it's trying to ensure that all these droplets come together on their own with the help of gravity, just hovering around, making sure we use the entire remainder of our media as well. Now, if we have a dry spot, we can just very gently guide the media on the edge of the device itself. And now that we have our media overlaid, we immediately want to move on to our incubation phase, because we know sperm, they like to move and they like to find edges and they like to swim up. So we want to immediately add the device to our Petri dish. This serves more than one purpose. Of course, this is a more tidy environment should we experience a spill, but it also will provide a lot more stability as we travel to and from our incubator within the lab. However, most importantly and primarily, what this is doing is retaining humidity. Again, we're only using 3/4 of a mil of media, so we don't want that to evaporate at all and risk any changes in pH. So we make sure we have the cover on the dish, and we move it to our incubator, set a timer for 30 minutes. And of course, we're not going to sit around and wait, since this isn't a real life situation, but you can imagine all of the other tasks or work that can be accomplished in that amount of time. Again, we know 30 minutes will provide, on average, the highest yield of sperm with the lowest levels of DNA fragmentation associated with them. So once that timer goes off, we want to capitalize on that moment, capture that moment. If we walk away, we'll know that other sperm will eventually find their way through. They may look the same, swim the same, but we won't know anything about their chromatin integrity. And of course, chromatin integrity, genomic competence, is the inverse of DNA fragmentation. So when we have removed the device from the incubator, I like to remove it from the dish as well. This way it's less likely to slide around on us, because at this point we've achieved a very special stasis here, where we have all of these motile sperm swimming in the media ready to collect on top of the device, while everything else we started with in the lower chamber of the device still. So we want to take our time, be mindful, and aspirate slowly, steadily, avoiding any turbulent motions or anything that could agitate the stasis that we've achieved. So you'll see me keeping 2 fingers on the device to avoid it sliding away from me or anything that we don't want. And from here, we'll place our third and final syringe into the outlet port for our final time. And we can aspirate a total volume from this device of 500 microliters, so 1/2 of a milliliter. And again, when we do so, just keeping our eye on things, using a steady hand, and making sure we do not over aspirate, as that will, like I said, kind of stir the pot, shake things up in a negative way. And once we have accomplished that, the device has done its job. We can dispose of it appropriately. And if it's ICSI or, like I said, any other time of the day we plan on using the sample, we want to immediately move it to our cap tube. Any tube will do, as long as it's a safe place to store sperm and that it has potential for a tight snap top or screw cap, such as this Eppendorf tube. And now would be the time that we take a test drop. Unfortunately, I can't show you that right now, But if you talk to anyone that uses ZyMōt, the word that comes out of their mouth when they take a look at that drop is wow, because everything is so clean. It should be void of, the sample should be void of any somatic cells, any debris, anything that does not have a flagellum should not be in our final sample. And that's one of the inherent major advantages of this device. Also with the added assurance, knowing that for ICSI, for example, when we're searching in our PVP or our sperm slow, and we are looking for that special sperm somewhat arbitrarily, we know that any and all are going to be, with ZyMōt, are going to be the ones that are equal potential. So just pick one that you like. So that is how ZyMōt works. And even in a situation like this with a lot of talking coming from me, I hope everyone can see that in a real life circumstance, this does not take very long at all.